So today I'm going to talk about chapter 23, which has to do with evolution of populations, population genetics. And it's important to talk about the evolution of populations because populations are the basic unit of evolution, not individuals. An individual cannot actually evolve. Only populations can evolve because evolution is definitionally the change in allele frequency in a population over time from one generation to the next. One example of this that we talk about in your book, and it's pretty famous, is an example about Darwin's finches. So on Daphne Major Island, which is one of the islands of the Galapagos, there was a drought. And a husband-wife team of scientists named the Grants had been measuring beak size in a large-beaked bird. And that particular type of finch, the average beak size went up over time during the drought over a couple of years. And the reason they discovered was because with the drought, there were only large, hard-to-crack seeds left over, and you needed a big bill to crack those. And so larger billed finches did better, and if you measured the average bill size in the population, over time it went up. So this is a look at the particular finch in question and a large seed that's trying to break. And as you can see, just over the course of a couple of years, before and after the drought, the average beak depth got significantly larger. The beaks got thicker so they could break larger seeds, and this is a change in allele frequency in a population over time, and it's an example of microevolution. So if microevolution is a change in allele frequency in a population over time, there are a few different things that can cause this. One is gene flow. If you get immigration of individuals from a different population that have a different set of alleles that can change your allele frequency, right? If you have a bunch of brown furred mice in one population and a bunch of black furred mice in another population. If the brown furred mice tend to immigrate into the black furred mouse population, that's going to change the allele frequency. Genetic drift can also change the allele frequency. This just means random chance. So if I have, let's say, 2 alleles in my body for eye color, right? I'm heterozygous. We remember what heterozygous means from last semester. Let's say I'm heterozygous for a particular trait. It's a 50-50 chance, right, that I could send the one allele or the other allele, right? It's entirely up to chance. Flip of the coin. And so chance plays some role, especially in smaller populations, in the gene frequency of a population. So genetic drift is that role of chance, and that can cause evolution. But the only thing that can cause adaptive evolution, meaning evolution that makes you more adapted to your particular environment, is natural selection, which we've been discussing. So one of the observations that Darwin made was that there was variation in heritable traits, right? And Mendel knew something about this. He was working on those units of heritability at the same time that Darwin was coming up with his idea of natural selection, though neither one of them knew what the other was doing. But now we know that the variation in heritable traits is caused by genetic variation, right? Differences in A's and T's and C's and G's, in particular segments of DNA that you find in individuals. Natural selection can only act on existing variation. So you have to have a way of getting variation into the population. Now, to be sure, not all phenotypes are caused explicitly by genotypes, right? We can have characteristics that are caused maybe more by the environment than by our genes, although usually there's some genetic component. Very often it's more than one gene that's involved, but as long as there's some genetic component, that trait is heritable. So where do we get this variation in our genetic codes? Mutation is the origin of all true variation, right? The only way we can get new genes, new alleles, is by mutation. And mutation is rare in one sense, but you make so many copies of so many genes, of so many A's and T's and C's and G's, of so many genes, that honestly it ends up not being that rare in any particular individual. So mutations produce this variation. They essentially produce new cards in the deck. But sexual reproduction helps you shuffle the cards in that deck, right? And that sort of exacerbates the amount of variation that you see. So sexual reproduction can cause variation in a couple of different ways. First of all, when you make gametes, every gamete you make is different. And that's because of the law of independent assortment, right? We have 23 different pairs of chromosomes. For every one of your chromosomes, right, you have one chromosome that essentially came from your mother and one chromosome that came from your father. And then those separate during meiosis. If you remember Bio 131, we discussed that. And just because one particular, gamete ends up getting your mom's chromosome 1, that doesn't mean that it's also going to get your mom's chromosome 2. It could get your dad's chromosome 2. Who knows? 50-50 chance every time. So that creates a fair amount of variation. Plus, we have crossing over during meiosis, which increases the amount of genetic variation by a huge amount. And then also, we've created a lot of different gametes, each individual. When two individuals get together, which gamete happens to fertilize which gamete from the female, right? Which gamete from the male and which gamete from the female come together is completely left up to chance. And so the number of possible genetic combinations in your offspring is just astronomical. In terms of mutations, which create that raw material for natural selection to operate on, it really is only mutations in the cells that produce gametes that matter for evolution, because those can be passed on to offspring, right? So, your skin is pounded with UV radiation from the sun every day, and that can cause and does cause lots of mutations. Those mutations can cause cancer if you get enough of the wrong mutations, right? But it really is only mutations that occur in the gametes, in the cells that are going to produce gametes, I should say. So within your testes or ovaries, that are going to be passed along to the offspring. And you could have a point mutation, which is a change in a single base in a gene that could cause a big effect or not. There are really a few options for each of those point mutations. It could result in change that is harmful. And in fact, that's often the case, right? And in that case, natural selection would weed that out pretty quickly, and you're not going to see it move on very far into the further generations. That said, beneficial changes, while they don't occur all that often, when they happen, they tend to spread pretty quickly in a population because they make you more likely to have offspring. In addition to harmful or positive, you can also just have neutral variation, right? You can have point mutations that just don't really cause any change, advantage or disadvantage, right? They may not even change which amino acid gets placed in a protein because there's some redundance in that code. And so neutral variation confers no selective advantage or disadvantage, although it can be interesting when you're trying to figure out how long two organisms have been separated, how long it's been since they've had a common ancestor. The more variation, neutral or otherwise, that they have, the longer it's probably been that they've been evolving independently of one another, the longer it's been since they've shared a common ancestor. Typically, deleting a bunch of DNA is harmful, or disrupting, meaning if you have a jumping gene that lands in the middle of another gene, that can be harmful. Duplication of DNA isn't necessarily harmful because you're not necessarily eliminating the function of another gene. You're just adding new DNA that may or may not do something else. In fact, duplicated genes, the duplication of entire genes, which happens pretty often, again, because of the jumping genes that we discussed last semester, can often be neutral or beneficial, and they can take on new functions by further mutation. So there's an ancestral odor detecting gene that has been duplicated a lot of times, right? Humans have 350 copies of it, and each of them have have changed a little bit over time and have a slightly different function. They've allowed us to smell a number of different things. Mice have 1,000 copies of that particular gene. They also have better olfactory sense than we do. While mutation rates per nucleotide are relatively low in plants and animals, and can be even lower in prokaryotes, although higher in viruses, because some viruses are RNA, which is harder to copy with fidelity. Sexual reproduction helps expand the amount of genetic variation that animals and plants have. That said, for prokaryotes, where you can't have sexual reproduction, they have to find other ways to expand their genetic variation. And the biggest way is rapid reproduction. So bacteria and archaea have very, very fast generation times. And so even if you don't get quite as much diversity generated in each individual generation, you have a lot more generations per unit time. Also, as we discussed in Bio 131, bacteria are able to transform some DNA from their environment, right? From maybe other dead bacteria, bring it into themselves. They're able to do some sharing of DNA through sex plasmids with other bacteria, and also sometimes viruses transduce DNA from one bacteria to the next, and so they do have ways of expanding that genetic variation. Anyway, we're talking about population genetics and the idea that populations are the basic unit of evolution. But if we're going to discuss this any further, we have to talk about what a population is. And it's sort of a difficult thing to define. It's a localized group of individuals that are capable of interbreeding and producing fertile offspring. So essentially, it's a group of individuals of the same species, right, that are close enough that they're able to interbreed. And that means we can treat them as a gene pool, right? We can talk about, for any particular gene, an individual only has 2 alleles, right? But the population may have lots of alleles. And because theoretically any individual could reproduce with any other individual, we can talk about the pool of genes in a particular population. For example, okay, these are 2 different, let me get this out of the way here, populations or herds of caribou, right? Now, they overlap geographically with one another, but they're constantly on the move, and our 40-mile herd range here and our porcupine herd range over here, while they overlap, the herds don't tend to be there at the same time, and so they don't do a lot of interbreeding between herds. So porcupine herd we consider one population, 40-mile herd we consider another population. Now, does that mean that there's never any gene flow between these 2 populations? No, it probably doesn't mean that. And admittedly, this can be a little bit of a wishy-washy thing. There can be some gray areas on what you consider a population. But the working definition of a population is just a group of individuals of the same species that will be able to share DNA with one another. Now, for some gene locus or another, it may be true that there's only one allele for that gene locus in a population. In that case, every individual in that population is homozygous for that one allele, and everybody has the same characteristic, right? But if you have 2 or more alleles for a particular locus, then individuals are not all homozygous. They could be heterozygous as well, right? Every individual has the possibility of having 2 different alleles for each gene locus in their genome, right? Now, you could have 3 or 4 or 5 or more alleles for a particular locus in the population, but any individual can only have 2. They can be homozygous, meaning they can have the same one twice, right? 2 copies of the same allele, or they could be heterozygous and have 2 different copies of 2 different alleles. In the next video, we're going to get pretty mathy. We're going to talk about the Hardy-Weinberg equations that describe the Hardy-Weinberg equilibrium. We're going to talk about how mathematically you can see whether or not there's been change in gene frequency or allele frequency in a population across generations.